Hello students, today we're going to understand one of the important cell organelles called peroxisomes. So, let's begin. Peroxisomes are tiny, round structures inside most animal cells, including human cells. They are very small, about 0.2 to 1 micrometer across, much smaller than the width of a human hair. Think of peroxisomes as special cleaning and recycling departments in the cell factory. Main jobs of peroxisomes. Number 1 function is that they break down very long fatty acids, fats that are extra long, longer than 22 carbon atoms. Normal mitochondria can't handle these super long fats well, so peroxisomes do a special version of beta oxidation, breaking fats into smaller pieces for energy or removal. 2. They deal with hydrogen peroxide, a harmful chemical. Many chemical reactions in the cell, especially oxidation reactions, create hydrogen peroxide, which is like a weak bleach. Dangerous if it builds up because it can damage other cell parts. Peroxisomes have lots of enzymes called oxidases that use oxygen to break down different substances, like some amino acids, uric acid, or fats. These reactions make hydrogen peroxide. But peroxisomes also have a superhero enzyme called catalase that quickly turns the dangerous hydrogen peroxide into harmless water and oxygen. That's why they are named peroxisomes, peroxy, from peroxide. Peroxisomes are named as such because they are peroxide-forming and peroxide-destroying bodies. The term was coined in 1966 by the Belgian cytologist Christian de Duve, who also discovered lysosomes. 3. They help detoxify, clean up, harmful substances that enter the cell or are made inside it. Liver cells have especially lots of peroxisomes because the liver has to clean many toxins from blood, like alcohol, megafats, etc. Inside some peroxisomes, you can see crystal-like structures. These are often big piles of the enzyme urate oxidase, which helps break down uric acid. How are peroxisomes made? They can split from already existing peroxisomes, like dividing bacteria. Or they can form brand new from vesicles that bud off from the endoplasmic reticulum or sometimes mitochondria. Most proteins that go inside peroxisomes are made on free ribosomes in the cytoplasm. These proteins have a special, address tag, called a peroxisome targeting signal. Helper proteins called peroxins read the tag and deliver the proteins into the peroxisome. What happens when peroxisomes don't work properly? If the genes for peroxins are mutated, peroxisomes can't form or function correctly. This causes serious diseases called peroxisomal disorders. The most well-known is Zellweger syndrome, also called cerebro-hepatorenal syndrome. Babies with this condition have problems right from birth: unusual face shape, serious brain, liver, kidney, and eye defects. Most don't survive long because peroxisomes aren't working. Very long-chain fatty acids build up and poison cells, especially in brain and adrenal glands. Another related condition is X-linked adrenoleukodystrophy, mostly affects boys. Here, the problem is mainly in breaking down very long-chain fatty acids. These fats pile up in the brain and adrenal glands, causes brain damage over time, worsening thinking, movement, vision, and adrenal gland failure, like Addison's disease. Low hormones from adrenals. In short, peroxisomes are small safety chambers in the cell that safely break down tough fats and destroy the dangerous bleach, hydrogen peroxide, made during those reactions. Without them working properly, toxins and bad fats build up and cause serious diseases, especially affecting brain, liver, and nerves. I hope you liked the video. Please subscribe this channel and press the bell icon.